everyone. In the previous videos, if you have gone through them, you might remember we are talking about how membrane transporters and channels and pumps function as machines to transport ions across the membrane. In this video, what we are going to look at how these different transporters, channels and pumps work together and interact with each other to regulate the cytosolic pH. As you see, there are so many reactions going on inside the cell, and because of that, cell has a slightly acidic pH compared to extracellular environment. Now, what is the importance of regulating proton concentration in the cell? Why does it need to maintain a pH in this acidic region? Most of the chemical reaction happens by exchange of protons. So proton exchange drives most of the physiological reactions, as we'll see. And then the surface charge dictated by protonation or deprotonation can play a role in protein function. For example, due to protonation, the conformational change in protein can drive its function. Along with it, as we have already seen, the electromotive force generated across the plasma membrane because of across the mitochondrial inner membrane because of the proton drives ATP synthesis. So if you remember, I had already talked about ATP synthesis, which uses the proton gradient to generate electromotive force and uses it to generate ATP from ATP. Now we are going to look at why this pH maintenance is important. Because it's slightly acidic inside, protons can move out of the cell across a gradient which we do not want. But also, a lot of reactions produce proton. For example, aerobic respiration in mitochondria produces carbon dioxide, which reacts with water and in presence of carbonic anhydrase forms carbonic acid, and this can reversibly break into a proton and bicarbonate ion. Now, this proton increase inside the cell will make it acidic and it can cause acidosis. And to avoid that, we have these transporters. For example, one of the main player of proton or pH balance is sodium proton exchanger, which takes the proton, and in order to make the exchange charge neutral, it exchange it for a sodium ion which has the same charge as a proton. So it brings in one sodium ion and effluxes out one proton. So this is an antipode. If you remember from the secondary active transport, this is an antipode where energy from sodium gradient is providing energy for the flow of proton. Now, how does it senses when there is enough proton that it needs to start working, or when the pH is low enough for it to start acting? Now there are two models for it, or two postulations. One says that there are some ionic residue amino acids or ionic side chains. Because they are ionic, they are charged, they can interact with proton, and the protonation or their deprotonation will dictate whether the protein is in an active state or not. And there are some evidence supporting this. But there is also another postulate which came from Monod and Wyman. Wyman. and Chingwicks. So this postulate says that there are two affinity states. If you remember the calcium pump as we had studied, there is one low affinity state and there is one high affinity state. And when there is enough proton present in the cell which is beyond a threshold, the the low affinity switches to high and it becomes activated. And these two models both are supported by experiments and they are not mutually exclusive. So one transporter can have both mechanism going on. Now we saw that the proton formed from this reaction is being sent out of the cells, but there is also generation of bicarbonate ions. And if the bicarbonate ion stays inside cell, that's also something we don't want. So in order to exchange it, just like the proton, we will want something charge neutral exchange. So for that, the cell exchange it for a chloride ion. As you know, chloride ion has same charge, one negative charge. And this happens via transporter protein called anion exchanger. Because this is exchanging two anions, this is called anion exchanger. So it brings in one chloride and in exchange it will send out one bicarbonate. So as you see, the product of the reaction from carbon dioxide with water, the product from the reaction is being removed, which means this reaction will be favored in a forward direction. So more and more carbon dioxide can also be expelled out. So in a way, this favors the cell in continuing aerobic respiration and it can expel out the carbon dioxide in the form of bicarbonate. However, there can be situations in which the cell want to bring in bicarbonate ion to maybe balance the proton or to balance the charge. And in this, or maybe for buffering the pH, because bicarbonate can be a good buffer. So there, sodium bicarbonate channels come into play and they bring in one sodium and one bicarbonate, keeping it charge neutral. So one positive charge, one negative charge. And as both are entering the cell, this is a sympode. The NHE and anion exchanger were antipode, but this sodium bicarbonate channel is in sympode. Now, while it brings, while some of them bring one sodium ion and one bicarbonate ion, there are some others, for example, NBC 1, 2, 3, and 1 will bring one bicarbonate, but 2 and 3 brings more than one. And then there is this sodium driven chloride bicarbonate exchanger, which is driven by sodium gradient, and it exchange bicarbonate with chloride ion. So as you see, there are so many of sodium entering the cell, and that is giving energy for the secondary active transport, because there is a sodium gradient, there is less sodium inside the cell and more sodium outside, so the inflow is providing energy. But what will happen if they keep functioning? You'll realize that the more and more sodium enters the cell, slowly the gradient will vanish. And to avoid that, as we had discussed, there is the sodium potassium ATPase, the sodium potassium pump, which through active transport is uses ATP. It breaks down ATP to ADP and gets phosphorylated. Oops. And through that, it drives out 3 sodium ion. So it will take 3 sodium ion and put them outside, and it brings in 2 potassium ion. And in that manner, it maintains the sodium gradient across the cell membrane. Now, this is what we saw what happens in aerobic respiration. But sometimes, let's say in muscle cells, when you are doing heavy exercise, when you need a sudden burst of energy and you don't have enough oxygen, they switch to anaerobic respiration where pyruvate breaks down to lactate and a proton, which is together lactic acid. And this lactic acid builds up, causes muscle cramps, if you know. Now, here you see there is an excess of lactate inside the cell than the outside of cell because lactate is being built up. And this high concentration creates a gradient, and using this gradient to give energy for proton transport, and this becomes a symport. And the key player for this is the monocarboxylate transporter. Monocarboxylate, which is like lactate, it's monocarboxylate transporter. Now, this monocarboxylate transporter MCT is mostly found in muscles, and you'll see its importance because in muscle cells which can undergo acidosis easily, 40% of recovery from acidosis or from cramps, 40% of the recovery happens because of this MCT, happens via MCT, monocarboxylate transporter. But as I have said, these are mostly found in muscle cells, skeletal muscles, which are actively working. So in most of the other cells, it's not really found. However, the other transporters that I have mentioned, like NHE and sodium driven chloride bicarbonate exchanger, anion exchanger, so those are very common. And at this moment, there are many of them known which regulate the cytosolic pH. The number can be more than 63. So there is a website where you can learn more about them and their subclasses. I'll put the website link in the description so you can check if you are interested about it. Now, up until now, we have seen proton being sent out of the cell or bicarbonate being sent out or brought in. But what if cell needs to get more acidified for some reason or other? Maybe for endocytosis it needs more acid or something. So in those situations, in these special cases where it might need to get more acidic, get protonated, it uses various mechanisms. One of them is a pump, ATP driven pump, which will break down ATP to ADP, and that will push out calcium out of the cell, calcium ion, and bring in proton. So this is called plasma membrane because it's on the cell membrane. So plasma membrane calcium ATP, because it is breaking down ATP, it is an ATPase. So plasma membrane calcium ATPase. It can also be happening that it just needs to push out calcium, so it exchange it for a proton. But in this manner, the cytosolic pH is maintained through the orchestra of all these different anion exchanger, cation exchanger, and pumps. In the next videos, we'll see how the pH of outside of the cell can be also maintained.